that these words make a lot more sense. Otherwise, it's just blah, blah, blah vocabulary, and who cares? Okay, so it all depends on all the colors. So this would be 2D. And what we're talking about is transcription. Okay, so you have your DNA, 5' to 3'. Just like always, that top right-hand corner is always 3', always. So then you get your other strand. Now, I'm not going to drop the bases in here because it's adding too much detail. I want you to have a conceptual understanding first, always. So if this is 5' here, this would be 3' here. DNA is always read in a 3 to 5' direction. It has to do with the way the sugars point, okay? The enzymes, whether it's RNA polymerase, whether it's DNA polymerase, whether it's RNA primase, whether it's the gyrase, whether it's helicase, whether they're transcription factors, they all read DNA 3 to 5, always, okay? So then you get your RNA polymerase in here. And get it there. So this is, oh baby, there we go. This is RNA polymerase. Now, I'll plant the seed right now. This is RNA polymerase 2. In IB, we do not get into RNA polymerase 1, but if you're a bio major, you will, okay? And then, as every other enzyme does, the RNA polymerase reads in a 3' to 5' direction, always. Okay? So then what is formed is complementary to this DNA strand. So here is your RNA, your messenger RNA strand, your immature or pre-messenger RNA, because you now know that this RNA is edited. Okay? Now, these bases here are complementary to these two bases here. Does that make sense? That this is complementary to that? Okay? Now,
yes.
If this is complementary to this, that means this was complementary to 3'. Since this is complementary to 3', which prime is the messenger RNA?
5'.
That's a 5, okay? So this is 5'. That is important because that means, I should have highlighted first, let's see if it blurs it. Not too bad. Because then when the ribosome goes to read the messenger RNA, which direction is the ribosome going to read this messenger RNA?
3 to 5.
Very tempting response. I'm going to say the ribosome reads 5 to 3. That's the only exception. Okay? DNA is always read 3 to 5. RNA is read 5 to 3. Why? Because the RNA is the complementary strand. It's the other side. Okay? So don't be confused by that. The reason why the ribosome reads 5 to 3 is because this messenger RNA starts on 5'. Why does it start on 5'? Because the messenger RNA that was formed in here is complementary to 3'. Okay? The strand that is being read, this is the template strand. The strand that is not read is referred to as the sense strand. Now, I consistently draw the bottom strand as the template strand because it makes my drawings easier for me, okay? But if IBO wanted to test your conceptual understanding, what would they do to this diagram?
turn it upside down.
They turn it upside down on you. Yep. And they would have the ribosome reading from here to here. Okay. If you're comfortable with that, awesome. If they have the ribosome up here and it makes you uncomfortable, take the exam and turn it upside down. And guess what? The RNA polymerase will still read 3 to 5. Okay. I consistently draw them on the bottom. Don't let an image of the RNA polymerase on top disrupt you. All right. Now, if you read this top strand, if you read this strand, then this strand becomes the template strand, and this is the sense strand. Whichever strand is being read is the template strand. Now, lately, there's 2 new terms that are starting to be used. They're starting to call the sense strand the coding strand. And the reason why they do this is this strand is complementary to the template strand, just like this messenger RNA is complementary to the template strand. Therefore, this strand here of DNA and this strand of messenger RNA are the same, except for thymine and uracil. And so they're starting to refer to this top strand as the coding strand because it has a very similar code to the pre-messenger RNA. Makes me a little uncomfortable because then students start thinking, oh, so the code is here. And you're like, no, the code is actually down here. You don't much care for the term, but if you happen to see it, that's what they're talking about. And then this bottom strand, rather than calling it the anti-coding strand, which makes sense, they're calling it the anti-sense strand. I really don't like this term. The original term that first came out, get a load of this, was they were calling it the nonsense strand. Ridiculous, okay? So you read the nonsense strand, like, oh, you've got to be kidding me. And I ignored it when it first came out. I refused to use it. I'm like, no, I'm not going to use it. And then about 3 years ago, finally, you started seeing anti-sense. It's still not a very good term. You don't see this very often. It's not rare, but it's really infrequent. However, if you get into a genetics class, a cell biology class, a biochemistry class as a bio major, and they start throwing around coding strand and anti-sense strand, that's what they're talking about, right? Most people still use template and sense. I don't see the need for these. To me, it's just, let's make up more terms. So let's step back and let's get a general idea of what is transcription. Because sometimes when we talk about this biochemistry, we get so distracted by all the details, right? The trees, that sometimes we don't step back and remember the whole forest, okay? You can't see the forest for the trees, so don't let the details bog you down. So what is transcription? 3 major things, okay? The whole point is you're synthesizing messenger RNA. Now, you now know that this is immature or pre-messenger RNA. Okay? The enzyme that does the work. This process, the synthesizing, is done by RNA polymerase. It's just a classic enzyme name, right? You read enzymes backwards, and so this is an enzyme. This is an enzyme that makes a polymer or a chain. This is an enzyme that makes a chain of RNA. Okay? And then I'm setting you up to understand that just like when I first introduced replication, and I said DNA polymerase 3, and you're like, what about the others? I'm like, later, and I've introduced some of those. This is the same concept. There's always more layers. Okay? And why is this process important? It's the first step in protein synthesis. Biochemistry of all cells is controlled by proteins. Therefore, the production of those proteins is extremely important. Anyone still need this? All right. Let's do who, what, where, when, why. So this would be 2D. The 5 W's of transcription. There's an apostrophe there, and there shouldn't be. Cool. Who, what, where, when, why. Who, what, when, where. My favorite word, why. I love that word. I got a little tired of it when Emily was about 2 or 3, but it's okay. It's not 2 or 3 anymore. All right, so who does the work? Piece of cake. RNA polymerase. Now, I will talk very briefly about how RNA polymerase is activated or inhibited, very similar to replication, where there is a primer that attracts the DNA polymerase 3. There are proteins that can attract RNA polymerase 2. They are called there are also proteins that repel RNA polymerase 2, and they are called inhibitors. And that's how cells turn genes on and off, are with inducers and inhibitors. But for right now, we won't even call them that. IB refers to them as transcription factors. And so I will mention them, and there's, of course, ready, more detail. So what's the whole point? This is the production of messenger RNA, okay? Now, all too often, students will say, why? Why have the messenger RNA as a go-between? Why not just go and have the ribosome directly read the DNA, okay? Why have the go-between? Very important, because this is a disposable copy of DNA. Okay, that is what makes messenger RNA so useful. Okay, the DNA stays protected in the nucleus, or in bacteria, it stays attached to the plasma membrane in the nucleoid region. It's protected. Okay, just like when you're in the regular class and your teacher gives you a copy, we never give you the original, right? We keep that to ourselves. Why? So I can just keep producing copies forever. Okay, so the whole role of messenger RNA is to be the disposable copy. You make a copy of the gene, you send it out into the cytoplasm wherever it needs to go, a free ribosome, an attached ribosome, whatever biochemistry has to happen, you keep your gene protected. Okay, when does this happen? In the cell cycle, this happens during the first growth phase, or G1, and during the second phase, G2. Okay, now a little of this happens during the S step, but very, very, very little in the S step. Why? Why would there be so little transcription during the S step of the cell cycle?
Because more DNA is being synthesized.
Yeah, because that's when DNA synthesis happens. That's when replication happens, right? Well, you can't have a RNA polymerase plowing through the gene while you're trying to have the concert of DNA polymerase 3 and 1 and ligase and all this other kind of highly coordinated, orchestrated biochemistry. Okay, so transcription dramatically drops during the S step. You still have a little bit, but I mean like a little bit, right? But not very much at all. Now, where does this happen? Well, if you're in a prokaryote, it occurs in the nucleus. But don't forget about those old prokaryotes, okay? It's called the nucleoid region. Nucleoid region, or the area in the cell where the DNA is attached to the plasma membrane. And then why? Why is transcription so useful, right? Because what it allows is it allows the original copy of the gene. It allows the genetic material to stay, can stay in the nucleus. Protected.
Miss Mel,
Yes, ma'am.
can you reiterate how you introduced the RNA polymerase 2? You were kind of cracky when you were explaining it.
Oh, sure. I'm simply implying that there's more than one RNA polymerase that functions in transcription. You don't have to worry about the detail right now. Just like when we first introduced replication, I called it DNA polymerase 3, right? Implying that that there were other DNA polymerases that we would talk about later. I've still only talked about DNA polymerase 1, and you won't hit DNA polymerase 2 unless you're a bio major in your cell class when you're about 20.
Oh, okay. Thank you.
Okay. You're welcome. Okay, so when we talk about the nucleoid region, I want you to be comfortable here. You have a prokaryotic cell, okay? You have the cell wall, all right? Then you have the plasma membrane inside, okay? And now what bacteria do is they anchor their DNA into, they literally plug it into, they embed it into the plasma membrane, and then they'll have this loop, and then they'll attach the other end to the plasma membrane. And so this is their, what is called genomic DNA, okay? And so this is then considered the nucleoid region, okay? Of course, because prokaryotes don't have a nucleus. So that's what we're talking about back up here. Okay. And then there's some images in chapter 17 of your textbook. Oh, let's see where they are. 17.8. And then we'll talk about 17.7a and 17.7. Let's get those pictures out. It's orange here too. There we go. Does anyone still need this? All right, so what we're going to do is we're going to get a little bit more specific on this general diagram of how RNA polymerase 2 attaches to the gene, reads the gene from 3' to 5', because don't forget DNA is always read from 3 to 5. Then the messenger RNA that is produced, the messenger RNA, since it's complementary to the 3' end, the messenger RNA is synthesized from 5 to 3, which means on Thursday, when we talk about translation, we will put the ribosome here, and the ribosome will read the messenger RNA from 5 to 3, okay? So what we're going to do is we're going to zoom in on these two strands of DNA with this image here. So this is 17.8. And so here is your, this is DNA here. Which prime do we always put in the top right? Always.
3'.
3'. Okay, that's your anchor. So if this is 3, that means this is 5. And if this is 5, that means this is 3. And if this is 3, that means this is 5. Always. Let's do that. Be efficient. I do them one at a time. Do them all at the same time. 3, 5, 3, and 5. Okay. This bottom strand is the strand that is going to be read. So this bottom strand is your template. Okay. All right. So there is an area where the RNA polymerase is going to bond, and then there's the area where the RNA polymerase actually starts to synthesize the messenger RNA. So I'm going to go forward just a little bit. So do you see how I made the messenger RNA here? Yes?
Yes.
Okay, I did that intentionally because so that right there, this, I think that would be great. Why do these little thin pins do that to me? This is my it's about to write a big R. This is pre-messenger RNA, okay? So even though the RNA polymerase is attached to this much of the gene upstream from the starting point, this is actually where the transcription starts. And so they put a little marker on it here. So that marker, that's where actually transcription starts, actual synthesis of messenger RNA. So then that marker, I really want attention to it, is there. And then that marker is there, okay? So then Campbell's puts in this little itty bitty arrow, right? And so this, this marker is the starting point, or some people call it the initiation point. Fine, that's where transcription actually starts, okay? However, you notice that the RNA polymerase, which to me always looks like a guitar pick. I don't know if any of you play guitar, but the enzyme totally looks like a guitar pick, okay? That it bonds to this entire area of the gene, okay? And so this entire area of the gene here, this is the bonding region for RNA polymerase 2, and this region where RNA polymerase bonds is called the promoter, or the promoter region. It promotes the beginning of transcription, okay? Now, inside of the promoter region, upstream from the gene, because the gene that's actually being transcribed is here. This is considered upstream. Upstream from the gene is a series of adenines and thymines, a lot of them, okay? And right before where the RNA polymerase bonds is always a thymine, an adenine, a thymine, and an adenine, always, T-A-T-A, in all species, not just humans, not just animals, not just eukaryotes, all living organisms on the sense strand of their promoter have T-A-T-A. It's referred to as the tatabox, and is one of the beginning sequences of finding genes. You search through the DNA, and there are certain sequences that act as binding sites, inducers, promoters, inhibitors, and so you search for these items biochemically, and that's the first step in trying to find the location of genes. So as you see this TATA, that is an indicator that that is the beginning of the promoter, and so we refer to this as the tatabox, okay? So the tatabox is upstream from the gene, and it is the very first place where the RNA polymerase 2 binds. Okay? Any questions on the starting point or the promoter or the tatabox before we move on? You guys good?
Yes.
Okay, so then we always talk about how cells turn genes on and off. Okay? There are little proteins that Campbell's lists here, and they made them kind of a light purple, and so I'm going to make them orange to draw a different color. I think it's a little misleading when they make the DNA blue and they come in with these proteins that are kind of purplish blue. So I'm going to make them orange. And these proteins function to either increase the amount of transcription, what we call inducers, or they work to shut transcription down, what we call inhibitors, okay? But whether these proteins are inducers or inhibitors, they impact transcription, right? So they are referred to as transcription factors. So these three guys here, these are called transcription factors. And these are proteins that will either activate the gene or shut it down. This is the switch of how genes are turned on and off by the cell. So do you think there might be a little bit of chemistry?
Yeah.
Yep. And then are there proteins that control the transcription factors?
Yep.
Yep. So even though these control the gene, these gene, but then there's controllers of the controllers, right? There's layers. It's awesome. It's not complicated. There's more detail that we can talk about now. Okay, and then this enzyme right here, then the enzyme comes in. So since the enzyme RNA polymerase 2 then shows up, that implies that these are inducers. So this is your RNA polymerase 2. Okay, let's get a nice green in here. And so this is your RNA polymerase here. And then as you get into more and more of your science, this is what cell biology and genetics and biochemistry is all about, is they'll hit these same topics, right? They'll go into transcription and translation and mitosis and meiosis and photosynthesis and cell reps. And they will simply give you more details as to how these processes work. That's what it's like to be a science major. Right? And then you have entire association of the RNA polymerase 2, your transcription factors, the initiation of your messenger RNA, this whole joining of all of these items together is called a complex. And so this is called a TIC, a transcription initiation complex. Transcription initiation complex. Okay. Any questions on that before we leave it?
All right, so there are some transcription factors behind the RNA polymerase 3 and in front of it. Are those still inducers or inhibitors?
No, those are controllers. There's another layer of transcription factors here. It's a nice eye, by the way, that will control the rate of transcription. It's a whole other layer. There's one layer of inducers and inhibitors that either draw in or repel the RNA polymerase. Then there's a second series of factors that control how fast the RNA polymerase will read that gene, whether it's slow, and so while the gene is being expressed, it's regulated, or whether it's induced, as in let's make a large quantity of this messenger RNA in a very short period of time. So this is another layer of control beyond initiation. Good question. Anything else? Thousands of bases a minute. Thousands, okay? And you do these presentations in 2 weeks. The animation that I'll show you today is super simple. I mean, like little kid simple, okay? Because I then want you to go out and jump into YouTube and find the modern 3D animations. They're amazing, and you will see how fast this works. It's machine gun. It's really fast. It's cool. Yeah, we draw it here like, A, U, G. It's not even close to being that slow. All right, then 17.7. All they're doing now is they're taking this picture here, right? And they're going to zoom in on this, right? They're going to give you more detail here. So that's what this is. Right? So when you come in here, they should get, no, I'll wait. This is, of course, RNA polymerase. This is your RNA polymerase 2. These are your RNA nucleotides. Okay. Now, since this is reading, right, this is your 3' here. So if this is 3, when this comes out, this is 5. Now, sometimes that messes with students because they go, man, come on, you told us top right was always 3'. As long as your strands are straight. Okay, what they've done here is, since this is being read here, this is being read 3 to 5. So this is 3, and that means when this is twisted, it's a 5 up here. So that's why it's different. And then this would be 3, and this would be 5. But the RNA polymerase is still reading 3 to 5. This is the direction of transcription. Of course, 3' to 5', of course. I should highlight that arrow so it really jumps out to you. Since this strand is the one that is being read, this is your template strand. What is being synthesized here, this is your pre-messenger RNA. Green crayon. And of course this is your RNA polymerase 2. And again, thousands of bases per minute. It's like a zipper, really fast. Just like that. So, 3-dimensional Star Tours ride. Could you be the transcription factors that float around in the liquid of the nucleus and then flow down and bond to the gene, right? And then big shadow as the RNA polymerase comes in here and binds, or you could write the RNA polymerase as it goes down the gene and the gene opens and closes. It's all right there for you. All right, so now what they're going to do is they're going to take this and instead of just showing it just sitting there, they're going to try to show you motion, right? Show you motion with a series of images here, right? So here's the binding, here is the initiation, here is what we call elongation, and then this is termination. So they're showing you step by step the process of transcription. So we'll go up here. Since these DNA strands are straight, there's no more 5' in the top right-hand corner. Okay, so 3, 5, 5, 3, 3, 5, 3, 5, 5, 3, 3, 5, 3, 5, 5, 3, 3, 5, 3, 5, 5, 3, 3, 5. Boom. Take it. So this down here, this is your DNA template strand. It's the strand that's being read. Okay. This is your RNA polymerase 2. So then from here back to here, this is your promoter. Okay. And then sitting right in here would be your data box. Nothing new here, simply different perspective on the same process as your RNA polymerase binds to your gene. Okay. Then there are these arrows here. Let's draw some attention to them. So as the messenger RNA, wish that was more red, so we'll make it more red. Okay, like pen. This is your pre-messenger RNA. Okay. And then your RNA polymerase is here. Since you are beginning the process, whenever you begin something, this would be initiation. Transcription is being initiated. As the RNA polymerase then progresses down the gene, the messenger RNA becomes longer. This is your pre-messenger RNA. Therefore, we refer to this process as being elongation. right? Begin the process of transcription, producing pre-messenger RNA. Continue the process there for your transcript. For this, your transcript is getting longer, fine. And then once the RNA polymerase reaches the end, there's an area here called the terminator. It's a whole different set of biochemistry. When the RNA polymerase gets to the terminator, the RNA polymerase physically changes shape and releases the gene, and the RNA polymerase releases the DNA and transcription stops. So I'll add some green color here and add some green color here. Cool. This would then be termination. And what you are then left with is your, pre-messenger RNA. However, since this pre-messenger RNA, this is complementary to this bottom DNA strand, which right there.
Can you repeat that?
Sure. Which time is right here. Since this transcript is complementary to this bottom strand, time is here.
5.
5.
Excellent. Excellent. That's a 5. Which then when we talk about translation on, let me think, Thursday, okay, when the ribosome attaches to the beginning of the messenger RNA, ribosomes read the mRNA from here to here from 5 prime. Cool pictures. Any questions on this before we show you a 1978 animation? Well, not 1978, but I'm intentionally using a simple one. Okay?
Yeah.
All right, so let's go over here. So here is your eukaryotic cell. Okay, so here's your plasma membrane. Here's your nucleus. Here's your double-stranded DNA, and they're showing you genes side by side. Okay, fine. All right. And the DNA is read through a process called transcription to produce RNA. Here is now your section of DNA where the RNA polymerase bonds with, thus the promoter. You know that the TATA box is in here. And then something we don't get into the specifics of is the terminator region here that causes the RNA polymerase 2 to release the gene. Right? And so then this is very straightforward. There's no helicase, there's no gyrase. RNA polymerase is left to do its own separating. Okay? RNA polymerase separates the strands. It then initiates transcription and linking RNA nucleotides together. And don't talk about now either. Do you catch that yellow spark? Consumption TP, when the nucleotides come in, they actually have some phosphates connected to them. Again, that detail is later. But the process here is relatively simple. The RNA polymerase 2 binds to the gene. It reads the gene, linking nucleotides together, forming your immature or pre-messenger RNA. And here they're showing you base by base with a little hint about the fact that ATP moves during this process. And as you asked earlier, it is not even close to being slow. It's really, really fast. Until the RNA polymerase gets to a section called the terminator, and that causes the RNA polymerase to release the transcript. Nope, come back here. I realized I had the loop on, just like that. And then what I would strongly suggest you doing is jump over to YouTube and type in transcription. There's what you're looking for. Okay. So this is a little bit more complicated, and then this is very, very specific. That's a good one right there. exciting as you watch. All right. So you are now qualified to watch all of these. So get in here, and some of them like this one, right? They show the ribosome like a little factory. That's adorable. Okay. Jump into and look at these 3D animations. They roll over and change all the time. It's really impressive. And support shown here in 2-dimensional static drawings. Get into YouTube and watch it fast. You will start to see how fast this process is, and watch it in 3 dimensions. Then we'll come back on Thursday, and we will then segue from transcription to translation. We will return to that good old drawing where we take the messenger RNA out of the cell and take the, or excuse me, out of the nucleus, and take the messenger RNA to an attached ribosome and a free ribosome. Pull the step back, see the forest. You got the forest, okay. And then we'll the second half of Thursday we'll drill down into the translate. Ribosomes at 9 bases wide. I only use 3 to get the concept across. And now we need to talk about the other thirds of the ribosome and how the tRNA comes in charged and then leaves. And we'll be done with 7 within about 2 weeks. Presentations, and then we'll 8, and then we'll do spring break, and you guys will be seniors, accept letters before you know it. Okay, so on your IA, don't forget to open up and start that Word document with all the different sections. The next Tuesday, you're turning in 2 documents. I want to see your spreadsheet. I want to see your data with your equations, and make sure that all of your equations are properly typed in before we start making conclusions on that data. And then we'll do topics 1 and 2 by next Thursday.